Hi everybody, welcome back to part 2 of the lecture series on synapses. Now that we know how a synapse stimulates a postsynaptic cell, we can now delve a little deeper into the graded potentials that we learned about earlier. And now we can see how the strength can be varied, and we'll even look at how one neuron can inhibit another cell, like a neuron or a muscle cell. Let's get going. Okay, so what synapses can do is, again, they can produce actually two different types of graded potentials. Okay, so if the presynaptic cell releases a neurotransmitter that opens a channel like sodium and depolarizes the postsynaptic cell, then that type of synapse is called an excitatory postsynaptic potential. EPSP. It's an excitatory synapse. And that's what you see on the upper graph there. It just means that, again, this type of neurotransmitter will open a gated channel that allows an ion to flow through that ends up depolarizing the cell. Okay, and most often the example we've been using is sodium. When sodium enters the cell, it makes the cell more positive and depolarizes it. But there are some neurotransmitters that can open different types of receptors or gated channels that will hyperpolarize the cell when it's open. So you can imagine these channels must be opening something like potassium. And remember, when a channel is open, potassium will flow out. And so that can hyperpolarize the cell. It could open chloride channel, which is negative. And if chloride comes in, then that's also going to hyperpolarize the cell. Okay. And so basically what we're seeing in this graph is that, again, the strength of the stimulus can change the membrane potential to a varying degree. Right? So the magnitude or the strength of the stimulus you can see below, that first one, the yellow one, is quite weak. And so that corresponds with the graded potential that has been produced above, and you can see it's pretty small. Okay? If the stimulus applied is a little stronger, then what we'll see is above it gets the potential actually rises a little bit and rises more toward threshold. Okay, if it's a very strong stimulus applied, then our graded potential can also be even stronger. The depolarization can be even more, possibly or absolutely approaching threshold, and then we'll get an action potential. So here's another way of looking at that. What is that different strength in the stimulus causing a different strength in the potential? So let's really understand this with our EPSPs first, or our excitatory postsynaptic potentials. So again, remember, they occur on the dendrites or the cell body, and that's what you see in the picture there. Okay. And if you look down below the dendrites or the cell body, you can see a stimulus is coming in, and that stimulus, again, is in the form of the amounts of excitatory neurotransmitter. So remember, an excitatory neurotransmitter is one that allows sodium in, or some other positive ion in, that allows the depolarization of the cell. Okay, and in this case, it was a weak signal. Maybe the neurotransmitter gate, the neurotransmitter only opened the gate for a short period of time, allowing only a little bit of sodium in, and you get that little red blip there. And that wasn't enough to reach threshold. Okay, so graded potentials themselves don't have a threshold. But what they're doing is, if they can accumulate enough, then they can cause the membrane to reach threshold. It's the action potential that has the threshold. And if it reaches that, it'll run. Graded potentials just means you're changing the potential of an area of the membrane. It's going to change. Okay? And it can either be strong or it can be weak. you can see in the second one that another stimulus came and it was a little bit stronger. And the third one, it was a stronger stimulus. So again, what does that mean? It might mean that again, two different neurons were stimulating this postsynaptic cell, and then their effects accumulated, meaning they summated on one another. So let's look what that means. We can actually see that there's summation that occurs. Before I do that, let me quickly show you the inhibitory, because here's an example of when a cell is being excited so that an action potential could be triggered. In this case, this is an inhibitory postsynaptic potential. And that means that the presynaptic cell then is releasing a neurotransmitter that is specific to a chemically gated channel that allows potassium to flow out of the cell or chloride to flow into the cell. So in both of those scenarios, the inside of the cell is becoming more negative. So we're pulling the postsynaptic cell away from threshold. Okay? It makes the membrane less excitable. That means if a stimulating or an excitatory potential comes, it's going to have to be even stronger in order to make up that difference to get to threshold. We've kind of talked about these concepts before. Now we're relating it to the actual mechanism that is causing the EPSP or the IPSP. Okay, so again, what's interesting about both EPSPs or IPSPs is that if there are enough of them on the dendrites or the cell body or both, then it can make the signal stronger. They'll accumulate, and that's what's making them stronger. So that accumulation is called summation. And there are two different ways we can get summation. So let's talk about the first one, known as spatial summation. Okay, spatial summation is illustrated here in these two pictures. Let's start at the left. What you're seeing is the cell body and the dendrites of a postsynaptic cell. And then all those little colored projections actually projecting toward it are the synaptic terminals of different presynaptic cells. And you know that they're different from the different colors. So those are each different presynaptic cells that can each behave independently. So each one of those can stimulate or inhibit the postsynaptic cell. Okay, and what you see on the right is what that would actually look like under a high-powered microscope. So if only one of those cells or one of those synapses on the left there fires and then it produces an EPSP on that localized region of the cell body of the postsynaptic cell, then it's going to produce a weak depolarization in that area, and most likely won't cause the depolarization to flow toward that axon hillock. And it's like that example I gave previously. It's like you standing at the end of the pool, the one side of that cell body, and you throw in one buoy or one floaty. It's likely not going to float to the other side of the pool. Okay? And so therefore, the axon hillock wouldn't reach threshold. But if you had several friends standing all around the pool, all throwing in floaties, then you're going to have more of them in there, and they're likely to flow to the axon hillock, reaching threshold. So that's the same thing. Each one of these synapses, those that are excitatory, meaning they have a neurotransmitter that opens a receptor on the other side of the cleft that allows sodium in, or another positive ion, then that membrane, those areas of the membrane, they're all going to allow sodium in. That's a lot more sodium flowing in. And again, they float through the cytoplasm. And if it causes the axon hillock to reach threshold, then we've stimulated this cell. Okay, and that action potential is going to flow down or be produced down the axon. Okay, so let's get the same concept, but we're going to look at it graphically. Okay, that's what we see here. So again, now what we're seeing in this graph are two presynaptic cells, number 1 and number 2, and they're both have a synapse with the postsynaptic cell, that third one sitting there alone. Okay, and so at the bottom of the graph, it's telling us at that first arrow that only the first presynaptic cell fired. That means it released the neurotransmitter. Okay? And that neurotransmitter on the cell body, or the dendrite of that postsynaptic cell, opened. Sodium flowed in, but you can see it only provided that little blip. It only went from -70 to what? Maybe -68. It wasn't very much. And so that's not going to cause an action potential. But now at the next arrow, it's showing you that not only did number 1 fire, opening up a chemically gated sodium channel, but so did number 2, which also opened a chemically gated sodium channel. So that's more sodium being released than only 1 could do. So the effects are additive. They summated. Okay? And so with that summation, that means the cell membrane depolarized and it reached threshold. This one's showing you -55. We've used -50 in the past. I told you it's somewhere between 50 and 55. Okay, that's enough to reach threshold, and our action potential now is firing on our postsynaptic cell. Okay, so spatial summation means that different neurons are producing depolarization on the same cell body. Okay, so it's across space. Different neurons in different areas are each depolarizing the membrane at the same time. So EPSPs from different synapses occur on the same postsynaptic cell at the same time. That is spatial summation. Okay, here's a little bit more example. Here you can see again, follow along. If only presynaptic cell A is producing an EPSP, look on the graph, we're not reaching threshold. Okay, EPSPs and IPSPs can actually cancel each other out, because now look at the red one labeled B. Okay, that one is, you can actually affect the depolarization of the presynaptic cell. So this one has an inhibitory neuron attached to that presynaptic cell A. So what that means is it's releasing a neurotransmitter that has a chemically gated channel that probably lets potassium out. And so it's causing the membrane in that area to hyperpolarize. So when the action potential coming down the membrane to that synaptic terminal, it actually cancels out the effect on the postsynaptic membrane. So B inhibited A. So you can see our graph there is flat. It remains at resting potential. The postsynaptic cell remains at resting potential. If you're confused in that graph, or we're looking at the membrane potential of the postsynaptic cell or the pre, read the y-axis. It says postsynaptic potential in neuron C. Okay. Look at D. D also produces an EPSP. It's not shown on the graph, but if both D and A fired, then those two would summate and it would probably reach threshold. But in this case, B is firing, once again inhibiting A. And so if both D and A fire, then you're only going to get, both D and B fire, then you're only going to get the effects of D, and that's not enough. So there's a lot of different scenarios that can play out. Alrighty. Just to go back here, just to show you that definition of spatial summation again. Okay, spatial summation has to do with you get several EPSPs because there are several different synapses occurring on the same postsynaptic cell at the same time. That is in contrast to temporal summation, so a different type of summation. This one occurs because the EPSPs that occur closely in time can summate before they fade. Okay, so this would be like one EPSP. So the green one, EX1 there, EPS1. You can see it's firing once, but its effects fade away before it fires again. So you don't get any summation. But now if it fires once and then fires immediately again before it's the first one's effect dissipates, you're summating. That means you're getting maybe twice the amount of sodium in that area. That could be enough to reach threshold. Okay, so some temporal summation happens, it's the timing. Temporal means timing. If one presynaptic neuron is firing very rapidly, then what's happening, it's releasing more neurotransmitter and it's keeping that those gates associated with that synapse, it's keeping those open longer, and therefore more sodium can flow in. So temporal summation has to do with how often the same presynaptic cell fires. And it can be happening in different ones. But if we see that several presynaptic cells are firing at the same time, like in this one or in this picture, right, then that's spatial summation. Okay, so spatial summation is referring to the space and several presynaptic neurons firing, and temporal is referring to how often a presynaptic cell fires. And so that can be illustrated in this picture as well. So this is a way of measuring action potentials, but it's measuring the frequency of action potentials. So on that presynaptic cell, in that first one, this would translate like if somebody was touching you really lightly. Your presynaptic cell then would be firing just maybe once and then again. So the action potentials would be being stimulated just once in a while. So that translates to less neurotransmitter being released. And so those chemically gated channels are just slowly opening and closing, allowing a little bit of sodium in. And so maybe once in a while you're reaching threshold. You are then interpreting that as a very light touch. But you can see if those action potentials come in more rapidly, then more and more neurotransmitter can be released, allowing those chemically gated channels to stay open longer. That's temporal, because we're talking about one action potential now increasing the rate that it's firing. And so you interpret it differently. Okay? So the thing to remember here, what we've learned from other lessons, is that APs are always the same strength, right? But they can influence the strength of the graded potential by increasing the frequency or how often they fire. Okay? And that's going to change the temporal summation. So make sure you can understand the difference between temporal summation and temporal summation and spatial summation. Okay, this last one, we're kind of putting it all together now. So notice over to the left side, we've got one neuron being stimulated by many other neurons. You can see the cell body of the postsynaptic neuron, but you can see several presynaptic neurons, their synaptic terminals, all merging on that postsynaptic neuron. That's referred to as convergence, right? Several neurons are converging on one neuron. Okay, so let me give you an example of what that might look like. So let's say that you are walking on the savannah and you see a lion. That's one presynaptic neuron bringing information. You hear a lion, that's another one, and you smell a lion. That's a third presynaptic neuron, and they're all firing on this motor neuron that's going to stimulate your skeletal muscles in your legs to run. Okay, so all of those senses converged on this motor neuron to get you to run. That's convergence. Now, in another example, one neuron can actually branch and stimulate several different neurons. Okay, it can stimulate many others. So that's called divergence, because the one neuron, its axons are diverging, and each of those collateral axons are coming into contact with a different postsynaptic neuron. Okay, so an example of that might be that you stub your toe. So that's one neuron bringing that information of the pain up to your brain. But then it branches, and so it sends a signal for you to pull your foot up, to jump up and down on the other foot, to cry, and to yell. Right? So that one signal of pain diverged and stimulated neurons to cause you to do those 4 different things. Okay? So that's divergence. Okay? So make sure you understand the difference between convergence and divergence. And so this is just explaining how our neurons are attached to one another in different ways so that you can get the many different responses that we have. Okay, so that video again gave us a little bit more insight now into the EPSPs and the IPSPs. Thank you.